Good afternoon, everyone, and welcome to my class today, which is related to membrane proteins and their function. So, I'll just give a quick summary or say recap whatever we have discussed in the last class. So, we were basically discussing about the proteins at the end of the day, right? What proteins are, what peptides are, proteins are polymers of amino acids, right? And we discussed some of the details related to how we can have a little bit of idea with respect to sequence. If we have a sequence, can we get some glimpse about what kind of a protein will it be, or will it be a membrane protein or non-membrane protein, right? So we studied about hydropathy index, which is one of the what to say benchmark parameter on the basis of which we check whether the protein has the potential or like the ability to be part of the membrane, right? And then we discussed about one such glycophorin protein, and we could easily see that the patch which is highly hydrophobic, that is, which has positive hydrophilic index, belongs to the residue which are highly hydrophobic, and they can be seen in the hydropathy plot itself, right? You can see that this is the stretch which I am talking about. If you look at the corresponding residues, these are the residues which are actually passing through the membrane, and they are highly hydrophobic in nature, correct? And remaining others, they have either very less positive hydropathy index number, or they have negative hydropathy index number, correct? So if the index is negative, it indicates that the residues are hydrophilic in nature, and their chances of being in the membrane will be very less. Okay. So I hope this thing was clear. So even in future, when you can see a stretch of amino acids which are highly hydrophobic in nature. Okay, let me have this uh, experiment. Maybe we can see if we can go to PTP, PTP. Mhm. Go to PTP, and we can see some transmembrane protein number. Let's say if we have potassium channel, right? So it's a channel at the end of the day. Channel indicates that it will be a transmembrane protein, right? It's a transmembrane channel at the end of it. Let's see this. Then it is a rich schema, or we can have, for example, let's see if we can see this one. Yeah, see, it's a protein. It is a membrane protein, right? We'll see the sequence. So this is the sequence, right? this much long, and these are the secondary structures. We haven't discussed about the secondary structures or structures of the protein at the end of the day, but this region is, and you can see the hydropathy plot of such section. So if we can zoom out a little bit with the sequence. Okay, let's focus on the sequence, right? So this is a membrane sequence, and you can clearly see that this is part of the membrane, and the residues which are commonly present here are mostly hydrophobic in nature. Okay, this indicates that we can have an idea about the protein property or structure, as well as the type of protein which it might be if we just look at the structure. Correct? And it is true only for the membrane proteins, because in membrane proteins a section of the protein needs to pass through the membrane, which is highly hydrophobic. So for hydrophobic region to accumulate proteins, the proteins and then amino acids associated in that particular region should follow hydropathy profile, or say hydrophobicity. Correct? So how many membrane proteins types are there? So we just studied that there are membrane proteins, right? There are different types of protein. There are membrane proteins, and there are different structures associated with it. We haven't discussed about the structures till now. We are only discussing about the membrane protein. So how many different types of membrane proteins are there? So if we look at the broad classification of membrane proteins, they are four in groups. Like first one is peripheral protein, then amphitrophic protein, integral protein, and then lipid linked protein. Okay, and this is the basic cartoon representation of membrane proteins. So excuse me. So if the protein has one polar part and remaining hydrophobic, chances are if this is the part, then it will be a peripheral protein. If it has a polar part and a stretch of hydrophobic part and then again a polar part, chances are this type of protein belongs to integral type of membrane protein, because they are the proteins which will transverse the membrane. Correct? So, and then we saw some of the basic of classification of membrane protein. So I'll just give you guys a little bit of refreshment again. Let me share the screen. Yeah. Yeah, so this indicates that the cartoon representation where the one part of the protein is polar, then a hydrophobic stretch, and then again a polar part. It's a characteristic of integral membrane protein. Okay. So I hope now that you guys have a little bit of idea on if you can see a protein, and so somehow you can give us an idea that what kind of a protein it will be, whether it will be a globular protein or a membrane protein. Okay. So before starting with today's class with the classification of protein, right? We're gonna handle each and every class of protein in the coming classes. But before that, let's have a quick video summary of the things which we wanna discuss today itself. Okay. Mm, yeah. right? So it was a basic introduction about the structures of protein. Don't worry, we're gonna discuss them in detail, but this basic information was necessary so that we can somehow get an image or idea about the type of secondary structures which will be present in the membrane. Okay. I have one more basic introductory uh, video which will help us in understanding our current session. Let me play that for you guys. Yeah. So these were the basic videos which I wanted you guys to see so that it will be easier for us to understand the coming lectures, because they are mostly theory associated. So I wanted you guys to have some basic idea about types of protein and their function, which we're gonna discuss gradually in our series, lecture series. Okay. So the first type of protein based upon this classification is the peripheral type of protein, right? So as I mentioned that this cartoon representation actually represent the peripheral type of protein. So what are peripheral proteins? Peripheral proteins are held to the membrane surface primarily through electrostatic or hydrogen bonds. They can be divided into two basic types, those that are attached through charges on the surface of integral protein and those that are attached to the anionic head group of phospholipids. Peripheral proteins can be removed easily and cleanly from the surface of membrane by either altering the pH or the media salt concentration. By definition, a true peripheral protein exhibits little or no interaction with the membrane hydrocarbon interior and is removed from the membrane without attached lipids. Classic examples of peripheral protein include cytochrome C, binds to an integral protein, and the myelin basic protein MBP, which binds to the membrane phospholipid. So, what it is saying that there are two different types of peripheral protein. One that gets attached to the integral protein, which we want to discuss in the coming slide. This will be a peripheral protein, and the one protein which is directly attached to the membrane lipid. It will also be a type of peripheral protein because it is associated with the periphery of the membrane, as the name suggests. Okay. Next type of protein is called amphitrophic proteins. So amphitrophic proteins are a relatively new class whose importance is rapidly becoming more and more apparent. Unfortunately, there is a lot of overlap between peripheral proteins that are bound to the membrane surface phospholipids, integral, ecto and endoproteins, and amphitrophic proteins. We're going to discuss this in literally the next slide. Okay, don't worry about this. Then amphitrophic proteins, they can alternate between conformations that are either water soluble or weakly bound to the membrane surface through a combination of electrostatic hydrophobic forces, right? So amphitrophic proteins, they can actually alternate between the water soluble and weakly associated with the membrane, while the peripheral proteins, they are associated with the membrane predominantly. Since the membrane interaction is reversible, an amphitrophic protein coexists as both a free globular protein and a membrane-bound protein. The water-soluble globular form is converted into the membrane-bound form following a conformation change induced by phosphorylation, acylation, or ligand binding that exposes a previously inaccessible membrane binding site on the protein. This is a self-explanatory point. So if a protein has to be membrane associated, there has to be some special type of residues, right? What we are calling here as hydrophobic residues. So if a globular protein is there and some changes are made to it, such as phosphorylation or acylation, due to which those hydrophobic sides of a protein are exposed, then they can get easily associated with the membrane. Membrane interaction is often based on production of hydrophobic loop of amino acid, sometimes an alpha helix that can penetrate into the lipid bilayer. Next, we have another important class of protein called integral membrane protein. Okay, so integral membrane protein, it is estimated that 20 to 30% of all cellular proteins are integral, and these proteins are tightly and permanently bound to the membrane. While peripheral and amphitrophic proteins are weakly bound to the surface of membranes and can be readily dissociated, producing lipid-free globular proteins, integral proteins are integrated into the membrane, hydrophobic interior, and can only be removed by more drastic means. Correct? Why? Because if we look at the representation, uh, in the video, if you guys remember, the integral protein will be associated in the membrane. So if you want to take out the protein outside the membrane, you need to provide energy to it, and this amount of energy, because you need to overcome the hydrophobic stable interactions which that protein has made with the membrane, and that amount of energy is very difficult to provide from the outside by changing the pH or salt concentration. Hence, integral membrane proteins are pretty difficult to extract and make crystals out of it in comparison to the other membrane proteins which are peripherally associated. Okay. Historically, integral proteins have been separated from membranes through acetone and other non-polar organic solvent extractions, cheotropic agents, and detergents. Although thousands of globular proteins have been crystallized and their 3D structures accurately resolved by crystallography, only a few dozen integral membrane proteins have been resolved. And you know the problem, right? The problem of obtaining 3D structures for the integral protein is in obtaining pure crystals in the first place itself. While peripheral proteins are obtained as monomers that are free of membrane lipid, integral membranes are isolated as aggregates with lipids, right, which are still attached to the protein and not in the pure form, unlike globular protein or peripheral proteins. The aggregates are almost impossible to crystallize. Therefore, excuse me. Therefore, structural information about integral protein is often obtained by indirect methods like enzyme degradation studies and hydropathy plots. There are two types of integral proteins, endo and ectoproteins. They do not transverse the entire transmembrane. Instead, they are inserted into the membrane's hydrophobic interior from either the cytoplasmic site, endoprotein, or the extracellular site, the ectoprotein. An example of such a protein is cytochrome B5, which is a part of EPS and it is associated with ER. Okay. Another important integral membrane protein class is the transmembrane protein. Now see how the videos were helpful, right? You can actually imagine in the last video you could see that there was first protein which was integral membrane protein. It was called monotopic protein. It was made up of single polypeptide chain and it will be a type of ectoprotein because it was present on the uh, extracellular side of the membrane, correct? Next is transmembrane protein. So a transmembrane protein, as the name suggests, it will be transversing throughout the membrane, right? So a transmembrane protein must span the entire membrane with segments exposed on both the outside and the inside aqueous spaces. The membrane that must be spanned is composed of a lipid bilayer that can be divided into three sections. The inner hydrocarbon region is approximately 27 to 32 angstrom thick. The very narrow boundary region between the hydrophobic inner core and the hydrophilic interfacial region is approximately 3 angstrom. Finally, the polar head groups region is approximately 8 to 10 angstrom. Okay, what it is saying? See, look here. If we look at the integral or a transmembrane protein, so these are the lipid bilayers, right? So the core hydrophobic region in which all the acyl chains are there, it is around 2.7 to 3.2 nanometer. Okay, then there is some region of around 3 angstrom, which is a very narrow boundary between hydrophobic inner core and the hydrophilic interfacial surface, right? And then we have the head group region, which is around 8 to 10 angstrom. So this is what it is saying, that a transmembrane protein, it needs to transverse this whole bilayer, then only it will be called as a transmembrane protein, correct? So it is rightly mentioned here. The very narrow boundary region between the hydrophobic inner core and the hydrophilic interfacial region is approximately 3 angstrom. Finally, the polar head, which is approximately 8 to 10 angstrom. Although this may be wider in membrane that include large amount of carbohydrate, right? So all membrane proteins are 100% asymmetrically distributed with respect to the membrane and do not flip-flop across the membrane. Therefore, transmembrane proteins are locked in a place. Characteristic of many transmembrane protein is the presence of tyrosine and tryptophan at the aqueous interface, which is this 3 angstrom long region. Okay, the amino acids serve as interfacial anchors that can interact simultaneously with the membrane hydrophobic interior and the aqueous interior Also, for reasons not entirely clear, the cationic amino acids like lysine, histidine, and arginine occur more commonly on the cytoplasmic side of the membrane. These are positively charged amino acids, and this is called a positive inside rule. This is referred to as a positive inside rule. The inner cytoplasmic leaflet is also the location of majority of the membrane primary amine lipids such as phosphatidylethanolamine and phosphatidylserine, indicating that PE and PS will be more commonly present in the lower leaflet or the leaflet which is facing the cytoplasmic side. Virtually all integral proteins have at least one stretch of approximately 20 hydrophobic amino acids that implies the existence of a transmembrane alpha helix. Alpha helix are found in all types of biological membranes and so represent a common membrane structure motif. Since each amino acid in an alpha helix spans approximately 1.5 angstrom, it takes 20 amino acids to cross the hydrophobic interior. Don't worry about this number. When we're gonna discuss about the secondary structures of protein, we're gonna see all these numbers. Okay. As expected, the transmembrane alpha helix are generally devoid of polar amino acid residues, while non-membrane spanning portion of the protein are usually enriched in the amino acids. Alpha helices can cross the membrane a single time or multiple time. So this is the basis of hydropathy profile also, right? Because the polar amino acids will be absent, and if a region is transversing the membrane, that region will be highly hydrophobic, and hence no polar amino acid will be present there. So there will be a stretch of hydrophobic residues in the sequence itself, through which we can get an idea that this protein might be a membrane protein or a transmembrane protein if the stretch is like 20 to 24 amino acid long, depending upon the type of cell and the thickness of the membrane. Okay. So today, I guess I'll stop here because it's a lot of classification coming on in the coming slides. So we have discussed, I'll summarize, we have discussed very little in the class, but still this is important because this will make the basis of certain uh, functionalities which are associated with the protein in the coming sections. So we have discussed about the peripheral protein. There were two different types of peripheral protein, one which is bound to the protein and one which has interaction with the lipid, right? And then amphitrophic proteins, which are, these are the proteins which basically uh, uh, like they are water soluble as well as membrane associated. For membrane association, there needs to be certain modifications like phosphorylation and acylations. Then integral membrane protein, we have seen if it is associated with the cytoplasmic side, it is called endointegral membrane protein. If it is associated with the extracellular side, it is called integral membrane protein. Then we have seen about the transmembrane protein. In the coming slides, we're going to see about different types of transmembrane proteins and some of the structural features, though you guys have a little bit of idea because of the videos which I have shared, but still don't worry, we're going to discuss about the secondary structures of protein pretty soon after discussing this classification of proteins. Okay, and then the last class will be lipid linked proteins which are associated with the membrane, and then we'll finally go to the structure of proteins. Okay, feel free to ask any questions or any queries if you have, and drop me a mail. Surely I'll try to resolve your queries. Okay, thank you so much. Bye bye.